In this video, we will learn about composition of blood. Blood is the fluid connective tissue, and this fluid matrix is known as the plasma. In this fluid matrix, it contains the corpuscles or the blood cells which are suspended. So these blood cells are of three types. One is the red blood cells, also known as the erythrocytes. Second is the white blood cells which lack the red pigment, that is the hemoglobin, hence they are white in color. Third is the platelets which help in the blood clotting process. So these three blood cells are suspended in the plasma. If we see the composition of the plasma, it is a pale yellow fluid which is part of the blood having 90 to 92% water, and rest 10% is formed of the substances which are dissolvable in it. For example, plasma proteins, mainly albumin, globulins and fibrinogen, which form 8%. Nutrients such as glucose, amino acids, fatty acids and glycerol, which form 0.1%. Respiratory gases such as oxygen and carbon dioxide. Excretory substances such as ammonia, urea, uric acid and creatinine, which will be present in traces, that will be less than 0.1%. Mineral ions such as sodium, potassium, calcium, magnesium. Chlorides, bicarbonates and phosphates form 1%. Enzymes, hormones and antibodies will be available in the trace amount. Next is the blood cells. Blood cells, also known as blood corpuscles, form the cellular component of the blood. It forms 45% of the blood composition, and there are three types: red blood corpuscles or erythrocytes, which transport oxygen. Then we have white blood cells or leukocytes for fighting against diseases. Then platelets, also known as thrombocytes, for blood clotting. Next, if we see the structural difference between these cellular components. RBCs, if we see, they are biconcave shaped. WBCs, on the other hand, are of many types. We can see here lymphocyte, monocyte, eosinophils, basophil and neutrophil, and they all differ because they all help us and provide immunity in different way. Then we have platelets, which are cell fragments of a larger cell. So first cell component that we will be discussing is red blood corpuscle, also known as erythrocyte. Cyte means cell, erythro means red. So this is a red colored cell. So these are minute biconcave disc-like cells. They are about 7.6 micron in diameter. Their cytoplasm is spongy and they are most numerous cells in the human body. Each ml of blood contains 5 to 5.5 million RBCs in man and 4 to 4.5 million RBCs in women. Now, what are the specialized features of RBC? If we take mammalian RBC, mammals are the ones which contain mammary gland, example humans. So in mammals, RBCs are highly specialized for efficient transport of oxygen. So how is it modified? If we see, it has the maximum amount of hemoglobin for transporting maximum amount of oxygen. And if we see the cell organelles, it lacks nucleus, mitochondria, endoplasmic reticulum, Golgi complex. All these cell organelles absence will provide more space to accommodate hemoglobin. So first, if we see, loss of nucleus makes the RBC biconcave, and this concavity will help in increasing their surface area by 30%. Thus, RBCs can absorb more oxygen. More RBCs can be accommodated in the given space. Next, since they don't have mitochondria, we know that mitochondria helps in production of energy, ATP, by breaking down glucose. Now, if mitochondria is absent, it will increase efficiency of delivery without using oxygen for their cellular respiration process itself. So without mitochondria, they cannot use oxygen. Thus, they will deliver the entire amount of oxygen to the cell, 100% of oxygen. And because of the small size, they can travel through fine capillaries. Elastic and disc-like body permits them to bend and twist as they pass through the capillaries. And red blood cells, we know it is a red color cell. The color is because of the hemoglobin pigment present. It is the oxygen carrying pigment. Hemoglobin readily combines with oxygen and forms a temporary compound known as oxyhemoglobin. So oxygen bound to hemoglobin is known as oxyhemoglobin. We can also denote it by HbO2. Now, RBCs laden with oxyhemoglobin will travel to the tissues in different parts of the body. There, they have to readily liberate oxygen so that tissue can instantly take it up. So, oxyhemoglobin will release oxygen. This will be taken up by the body cells and hemoglobin will circulate back to lungs. Now, hemoglobin also combines with small amount of CO2 and forms carbaminohemoglobin and brings it to lungs from the tissue, so lungs can throw it out of the body. Now, sometimes hemoglobin can also bind to another gas, that is carbon monoxide. And carbon monoxide can bind more strongly compared to oxygen and carbon dioxide. So we can tell that hemoglobin has greater affinity with oxygen but has 250 times greater affinity with carbon monoxide. So it forms a much more stable compound and this is known as carboxyhemoglobin. Now if you give a chance between carbon dioxide and oxygen, the probability of it binding to oxygen is more. So we can tell affinity towards CO2 is less. But if you give a chance between carbon monoxide and oxygen, it will have less affinity towards oxygen. So highest will be carbon monoxide and lowest will be carbon dioxide. Now if carbon monoxide bound hemoglobin, that is carboxyhemoglobin, is present in the bloodstream, it will cut down the availability of hemoglobin for oxygen binding and thus reduces oxygen transporting capacity of the blood. Now, shortage in availability of oxygen causes unconsciousness and leads to death, and this is known as carbon monoxide poisoning. Now, if we see the formation, lifespan and death of RBC, the formation process is known as erythropoiesis. In an adult person, about 2 million RBCs are formed per minute in the red bone marrow of flat bones such as ribs, breastbone, etc. Also in the end of long limb bones. Whereas in embryo, RBCs also develop in liver and spleen. And the process of formation or the production of RBC is known as erythropoiesis. During maturation, nucleus, mitochondria, endoplasmic reticulum all will be lost. So mature RBC will lack cell organelles, whereas immature RBC, which are still forming in the red bone marrow, will contain these cell structures. Now vitamin B12 and folic acid are essential for the maturation of RBCs. If we see the lifespan, so since they do not have nucleus, mitochondria, endoplasmic reticulum, they have a very limited lifespan. So the lifespan will be approximately 120 days, that is 4 months. Now death of RBCs is known as hemolysis. Lysis is breakdown. Hemo means hemoglobin containing cell. Now the breakdown of old and worn out RBCs is called as hemolysis. It occurs in two organs, that is spleen and liver. Now RBC contains hemoglobin and this hemoglobin can be broken down and some parts can be reused. So iron released during the breakdown of hemoglobin is retained in the body and it will be reused in the bone marrow in hemoglobin production. And the breakdown of protein part, that is the globin part, forms a pigment known as biliverdin, which is transported to liver. Here it gets changed to another pigment, that is bilirubin. So this will become part of the bile secretion from the liver, and it will be removed through undigested food. Next cell is white blood corpuscles, also known as leukocyte. White blood corpuscles are nucleated, irregular amoeboid cells. Their number varies from 5000 to 9000 per ml of blood. The ratio between RBC and WBC is 600:1. Now, WBC concentration can increase during infectious stage. Their number will be lower when we are not infected. If we see the structure of WBC, these are colorless, rounded or irregular shaped cells. They move like amoeba. That means they can change their shape and move through narrow capillary between epithelial cells. So they may even come out of the capillary, and this phenomenon is known as diapedesis. Now WBCs are of many types. We have granulocytes, we have agranulocytes and granulocyte. Now if we see the lifespan, WBCs survive anywhere between 12 hours to 12 days. The neutrophil type of WBCs can survive only for few hours. About 125 billion neutrophils are produced each day. And the old and worn out cells are destroyed in liver and spleen again, and the process of formation of WBCs is known as leukopoiesis. The new WBCs are produced in bone marrow, lymph glands, spleen and thymus. If we see the types of leukocyte, we know that they are divided into granulocyte and agranulocyte. Granulocyte because the cytoplasm of these cells contains granule-like structure and an irregular and many-lobed nucleus. So we call it as polymorphic. Here we can see, so eosinophil has a two-lobed or headphone shaped structure, that is nucleus, whereas basophil has three-lobed nucleus and neutrophil has 3 to 6 or multi-lobed nucleus. Now each of them have their own function. First, if we take acidophils, also known as eosinophils, they form 1 to 3% of total leukocyte. Their number increases during infection and allergic reaction. And the nucleus, as we see here in the picture, it is bilobed, and cytoplasm has granules which are large and alkaline. Now when we stain this granules, they get stained with acidic dyes such as eosin, hence their name is acidophils or eosinophils. Now the granules contains toxic chemicals which kill large parasites and detoxify bacterial toxins and promote tissue inflammation. Inflammation refers to swelling of a tissue, so more blood can flow through it and WBCs can reach faster. Next cell is basophil. They are about 0.5 to 1% of the total leukocyte percentage. Their nucleus is three-lobed nucleus and cytoplasm has acidic granules, and these granules get stained with basic dyes, hence the name basophils. The cytoplasm contains heparin and histamine, very important compounds. Heparin is an anticoagulant, which means it prevents blood clotting within the blood vessel. So imagine blood is flowing through the blood vessels and randomly if it clots, it can stop the blood flow. So heparin helps in smooth blood flow within the blood vessels. Whereas histamine is an inflammatory agent. As mentioned earlier, it will help in swelling. It is released in allergic responses and in injured tissues to dilate blood vessels and increase the blood flow. Next is neutrophil. These are the most numerous, forming 70% of the total leukocytes. Their nucleus is 3 to 5 lobed. Granules are large and are stained with neutral dyes, hence the name neutrophil. They are phagocytic in nature, that means they go and engulf the pathogen. So it's a cell eating process. So they engulf and They will kill the cells or the pathogens inside their body. They undergo phagocytosis, remove cell debris, so hence they are scavengers. They can squeeze out through capillary wall. The process is known as diapedesis. Next is agranulocyte. As the name says, A means absence. So the cytoplasm is without granules but has a large rounded or kidney shaped nucleus. Here if we see, lymphocyte has a large single spherical nucleus, whereas monocyte has a kidney shaped nucleus. Now if we see the first type, that is the lymphocyte. Lymphocyte is the smallest leukocyte and forms 30 to 35% of total leukocyte count. It will number about 1800 to 3000 per ml of blood. Now the cells are rounded with large rounded nucleus and very little cytoplasm. These are important cells because they are the ones which produce antibodies and provide immunity against infection. On the other hand, if we see monocyte, they form 3 to 7% of total leukocyte and they number just 200 to 700 per ml of blood. So very difficult to observe under microscope because of their less number. The nucleus is large kidney shaped or horseshoe shaped. Like neutrophil, monocytes also help in phagocytosis, that is they engulf microorganism and worn out tissue cells and keep the organ or the tissue clean. So they are also called as wandering cells of the blood. Next is the third type of blood cell, that is the blood platelets, also known as thrombocytes. Platelets are small, oval and spherical non-nucleated disc-like structure. That means they do not have nucleus. They are basically fragments which are derived from a cell known as megakaryocyte. Mega means big, karyos mean nucleus and a cell. A cell which had a larger nuclear structure broken down to produce these fragments known as platelets. So these are the fragments of megakaryocyte, which was a undifferentiated giant cell in the red bone marrow. They are 3 to 4 micrometer in diameter and they will be around 200,000 to 300,000 per ml of blood, and they survive only 5 to 9 days. And the old platelets will be destroyed in spleen by macrophages. I hope it is clear to you. Thank you.